Bismillahirrahmanirrahim. Hi, we're a team from University of Brawijaya. My name is Randy Zafira as a presenter, and this is our team. Here we are to present potential of Dregs to interferences Staphylococcus Aureus cell communication toward biofilm synthesis to support grain industry. According to Statistic Indonesia Plantation 2017, tea farms in Indonesia is quite extensive, 53,009 hectares. Tea production remains stable among these 5 years. Unfortunately, the export volume has been decreased. We know recent years' attention has been shifted towards the materials which produce less byproducts of wastes. Consequently, production with eco-label has become more important, especially to expanding the global tea market. The large production of tea as well as the resulting waste, the availability of tea waste is high, with a total production of 166,000 tons per year, and is currently not widely used. Staphylococcus aureus is one of the pathogenic microbes that contribute greatly to cases of food poisoning in the world, especially with its ability to form biofilms in food industry equipment. The presence of this biofilm can reduce the effectiveness of sanitation, contaminate food products, and can increase the level of antimicrobial resistance. Usually, food industry using chemical sanitizers to force biofilm. Unfortunately, it's dangerous because of potential entry of the residues into the product. The other way is using enzymatic handling, but it is expensive. Therefore, it is necessary to have a new strategy in overcoming the problem. Previous studies reported there is a way to force the biofilm by interference to quorum sensing receptor. This mechanism then applied to this research. The objective of this research was to investigate whether tea solid waste could be used as a quorum sensing inhibitor of Staphylococcus aureus pathogenic bacterial cells in biofilm synthesis. The following pictures are the materials that we use in our research. Our method is divided into 4 treatments that use different tools and materials, starting from the first step. It's about tea pulp preparation and extraction. The sample was taken from the tea industry waste that we use only 50 grams, dissolved in 400 ml ethanol 60%, and extracted using microwave-assisted extraction for 8 minutes. The next step is bacterial isolation test and media preparation. Staphylococcus aureus was inoculated to the Luria bertani medium on the first day and HDR media for the second day in the suitable condition, using incubator for 24 hours and 37 degrees. And then the third step is cell attachment inhibition test. In this test, we want to find out whether Dregs could inhibit the attachment of Staphylococcus aureus bacterial cells to the microplate. Therefore, we use different concentration of Dregs in 25, 62.5, and 100%, then pour into the microplate and rate the optical density using the microplate reader, then calculate it using this formula. The last step is the qualitative test of quorum sensing system inhibition. We use petri dish containing a mixture of media and Dregs with different concentration, and then put the Staphylococcus aureus culture. We can identify an activity from exoprotease enzyme that can be seen from the presence of clear zones around the colony. From this table, we can obtain qualitative data on Staphylococcus aureus quorum sensing activities. Next is result and discussion. This video shows about biofilm formation. Biofilm occurs in a series of steps such as formation of conditioning layer, bacterial adhesion, bacterial growth, and biofilm maturation. Most of the Staphylococcus aureus strains form the biofilm in an ICAA-dependent mechanism. Staphylococcus aureus strains that have at least one intercellular adhesion gene involved in the formation of polysaccharide intercellular adhesion, or PIA. This gene can synthesize PIA, which has an important role in cell aggregation and extracellular polymeric substance formation. Biofilms are formed as a result of quorum sensing from bacteria, where it secretes a molecule into its environment and then becomes a signal for the bacteria itself and other bacteria. This signal called an autoinducer. When the concentration reaches the threshold, the signal diffuses back into the bacteria and regulates the transcription of specific genes. This This picture shows about cell density-dependent gene expression in quorum sensing. It is a system to stimulus to coordinate gene expression with other cells and response related to the density of their local population. During quorum sensing, signaling molecules attach to the receptors of new bacteria and help in transcription of genes within a single species of bacteria, as well as between different bacterial species. Many ways that we can use to eliminate biofilm, such as using anti-biofilm. The main mechanism anti-biofilm compound is degradation process of polysaccharide components and biofilm matrix, inhibiting alarmone, disrupt cell signaling or quorum sensing, interfere with the regulation of genes responsible for the formation and transport of binding genes. This picture shows about inhibition of Staphylococcus aureus biofilm. More than 80% of flavonoids cause less than 40% inhibition of the biofilm formation by Staphylococcus aureus at 400 micromolar, and they were declared inactive. Phenolic compound has proven effective in inhibiting the growth and biofilm formation by pathogenic bacteria. Some phenolics, such as flavonoid, can interference communication signals of biofilm-forming pathogenic bacterial cells through the allosteric inhibitory mechanism in quorum-sensing bacterial receptors. Flavonoids work by binding to one of the adhesion proteins in bacteria. They inhibit the intercellular adhesion genes of ICAA and ICAD, which has responsible to synthesize polysaccharide intercellular adhesion. The conclusion from this research is phytochemical compounds from plant extracts, tea dregs, especially tannins and flavonoids, have the potential to inhibit the growth of biofilm formation in Staphylococcus aureus bacteria. This compound can inhibit the intercellular adhesion genes of ICAA and ICAD, genes that can synthesize polysaccharide intercellular adhesion, which has an important role in cell aggregation and EPS formation in the process of biofilm formation. Thank you for your attention.